Good morning, dear students. I'm going to continue the rest of the topic, the continuation of semi-conservative DNA replication. You already know that semi-conservative DNA replication have a property where both the parental strands involve in production of a complete new strand, that is daughter strand. Soon after the completion of DNA replication, the newly formed DNA strands have one parental strand and the other one is daughter strand. So the black one that I have represented is a parental, the newly formed one is a daughter strand. So this is called semi-conservative DNA replication. In order to prove the fact that DNA undergoes semi-conservative DNA replication, Matthew Meselson and Franklin Stahl had made an experimental proof by taking few things like this. Number one, they used the radioactive isotope of nitrogen, preferably N15 and N14. These nitrogen N15, when it is undergone centrifugation, it is more heavier than the N14. Okay, so when centrifugation is done, any chemical or any organism having nitrogen in it, especially N15 in their body, will obviously settle at the bottom of the test tube after centrifugation compared to N14. Second thing they did was they have taken E. coli bacteria. E. coli bacteria is the bacteria which have capability to undergo replication. It's DNA replication. Suppose if you take the DNA of that bacteria in order to produce a new DNA, it will take 18 minutes. Whereas the whole body of bacteria to undergo binary fission to form two, it will take 20 minutes. Remember the timing. 18 minutes is for DNA replication inside the body, and to divide into two, it takes around 20 minutes. So soon after 18 minutes, the 2 minutes left is for binary fission. So the complete DNA replication, that is karyokinesis, not karyokinesis exactly, DNA replication takes 18 minutes. Soon after that, cytokinesis follows, where the body divides into two. So it will take 20 minutes. Okay, now let's move into the real experiment. So I'll give you a clear cut and easy way to understand. Now imagine taking a test tube full of bacteria in the nutritive medium consisting of bacteria like this, and keeping or pouring the whole bacterial content into the medium consisting of N15 containing NH4Cl. A medium is nothing but the environment for the growth of the bacteria. Suppose the place where we are living, Earth, is having all the required things that are meant for survival of an organism. Same way, a petri dish or a test tube having a medium consisting of all the growth elements, all the required things for the bacteria to survive and also undergo multiplication. So the whole medium is transferred, this whole medium is transferred into the medium consisting of NH4Cl. Okay, now listen. When these bacteria has been left inside this N15 containing NH4Cl medium, the bacteria will multiply. Obviously, if you leave it for a few hours, say something like 4 hours or 5 hours, the whole bacteria will multiply while they are replicating. That means you know that when they are multiplying, they need to produce an extra DNA. So while they are multiplying, when their DNA is undergoing replication, suppose this is the DNA inside the body of the bacteria. So in order for the formation of a new DNA, Obviously, DNA consisting of nitrogen in the form of nitrogen base. You know that, right? Adenine, guanine, thymine, cytosine, right? So these, in order for the formation of a new nitrogen base, they take only the nitrogen source, that is N15. So after a few generations, after 5 or 6 hours, you find that bacteria, both the strands of the bacteria consisting of N15 medium, you can see N15 nitrogen in their DNA, because the only source available for the formation of new strand of DNA, in order to form new strand of DNA, it uses the N15 medium, because there is no such facility is available, because we are, or the Meselson and stahl has grown the bacteria in only medium consisting of N15. So obviously the newly formed strand will have N15 nitrogen base containing DNA. So after this, you can see the DNA inside their bacterial body have this kind of a makeup. Now, in order to make sure that whether the DNA is uh, having N15 medium, simply by doing the centrifugation will conclude that, because when the centrifugation is done by taking all the nutritive medium into the test tube and do the centrifugation, you'll find all the content is settling at the bottom of the test tube. That means N15 medium isotope is present in the whole nutritive medium. Thus, we can confirm that the bacteria present inside them have the DNA having N15 nitrogen base. Now, the real experiment starts here. Now, take this bacteria, taking one bacteria or millions of bacteria, say, take these bacterium into and transfer them into N14 containing medium. So once it is transferred and leave it for 20 minutes, you know that why we have to leave for 20 minutes, so that the bacterium will multiply and even replication also completes. So when it is getting multiplied during the replication, see, the DNA that previously the bacteria contain is both the strand having N15 and N15, both the strands are having N15 isotope. So when you transfer it into N14 medium and leave it for 20 minutes, what happens is both the strands, you know that they undergo semi-conservative DNA replication like this. Same way, this N15 both strands will need to produce a new strand. So in order to produce new strand, they have not N15 medium, because they are grown in N14 medium. So they take only nitrogen source, that is N14, to produce a new strand. So it will look like this. So this strand will have a newly synthesized strand having N14 medium. Same goes to this also. Here, this is how it looked like after 20 minutes. That means the DNA looked like this, because in order to synthesize a new strand, these are new strands, the only nitrogen source available is N14. So I'll write here N14 newly synthesized strand, and the red one is a N15, because this old strand is coming from the bacteria having, which is grown in N15 medium. So according to semi-conservative DNA replication, you know that newly synthesized strand will have one parental strand, that is N15 strand, and newly synthesized strand, which is N14 strand, because we had, or the scientist had grown that bacteria in N14 medium, the only source with which the bacteria uses to produce a new strand. Okay, so here same thing, N14 strand and N15 strand. Okay, now if you keep this whole bacteria to be grown for another 20 minutes, same thing, same, keeping the medium same and let the bacteria to undergo replication for another 20 minutes. So that is up to 40 minutes. What happens is that, see, this DNA has to produce 2 more DNA, semi-conservative DNA. So what happens is that this DNA, this strand, especially this N14 strand, need to produce, so I'll draw both the strands here, this strand here. So N15 strand of one of the parental strand. Okay, so now what happens is, after 40 minutes, going further for another 20 minutes, they have only N14 nitrogen medium, because we didn't change the medium. So N14 will be used to produce an extra new strand. So the newly synthesized strand will have N14 medium, and this strand, the parental strand, will have an extra N14 nitrogen containing strand. This is the parental strand. This is N15 strand. Okay, so same way, this specific DNA also need to undergo replication. So here I'm drawing the N14 parental strand and N15 parental strand. So this is the N15 parental strand. So after 20 minutes, same thing happened, where it uses N14 medium to produce a new strand. So both the strands will be N14, and this strand will have N14 strand. One is, this strand is N14 and this strand is N15. Okay, now listen, in order to confirm whether the DNA have N14 or N15 medium, like we did here, when we grown all the bacteria with the N15 containing, both strand containing N15, they weighed heavy, so they settled at the bottom. But when you take this same thing and do the centrifugation, see, it's very easy to find. Some of the bacteria will be, sorry, some of the bacteria, sorry, most of the bacteria will be in the neither at light stage or heavy stage, but they are settled at the middle of the test tube, because the reason is because they have both N14 and N15 containing strand. Because like I said, the N15 is heavier, so having N14 makes it not heavier, not lightweight, but the intermediate or hybrid. Okay, same thing, if you, in order to confirm it, if you can do this, same thing will happen. They'll settle at the middle of the test tube. Even they have represented graphically here, you can see. See, now what happens in this experiment is different here, not experiment, sorry, this generation is different. What happens here is, some of them are having N14, N15 strand, but you can see where they are present, N14, N15 strands here and here. But there are other bacteria which can settle at the top of the test tube. These are purely N14 medium containing bacteria. So because they have N14 and N14 strand, you can see these two, this and this strand. So this centrifugation is to confirm what? Confirm the settlement of bacteria based on the density. Now my homework for you is that, what if, you have to tell, what if this bacteria, that means suppose this bacteria containing this intermediate or hybrid DNA is grown in the medium of N15, the medium of N15, NH4Cl. So you need to show me what happens after 20 minutes. See, again I'm repeating the question. If you are growing this bacteria having this hybrid DNA and keep it for 20 minutes under N15 containing medium, what could be the fate of the bacteria DNA of that particular bacteria? And even some of you can do the same by taking this N14, both strand N14 DNA bacteria, and grow it in N15 containing medium. After 20 minutes, what could be the DNA structure? You have to tell me. This is your homework. Now, so this confirms that DNA undergoes semi-conservative DNA replication. It's very easy and simple to understand also. Now, this experiment was done on E. coli. E. coli is a prokaryotic bacteria, right? This is a prokaryotic. In order to confirm whether the same thing happens, that is, same semi-conservative DNA replication happens in eukaryotes, there were some other scientists. Here I have written here, or I've taken. There are some scientists, Taylor and colleagues, in 1958, done the same experiment, semi-conservative DNA replication, but instead of doing it on the E. coli bacteria, they did on a eukaryotic plant. Obviously, we all belong to eukaryotes, na? So, in order to prove the same, whether the semi-conservative DNA replication happens in the eukaryotes or not, Taylor and colleagues in 1958, they had taken a specimen, which is a plant, which is called Vicia faba, a plant which is a eukaryote, done the same experiment, and they also concluded that even eukaryotes DNA will undergo semi-conservative DNA replication. Remember the scientists. So what they did was they used radioactive thymidine. Okay, instead of using nitrogen N15 or N14, they used radioactive thymidine. So they concluded the same. So you can see the experiments proved that the DNA in chromosome also replicates semi-conservatively. So this is what all about semi-conservative DNA replication. And I'm leaving you with the homework. So please make sure that take a screenshot and then try to solve what happens if these two strands is grown under N15 medium. What could be the result after 20 minutes?